Question 4. The neurotransmitter, I'm better just saying GABA, binds to GABA-A receptors in nerve cells, and GABA-A receptors are a family of structurally related transmembrane ion channels. One proposed structure of a GABA-A receptor is shown in the diagram. It's quite a nice picture showing a nice set of alpha helices in there. And then obviously an extracellular receptor point on the outside. Okay, all GABA-A receptors consist of 5 subunits. Name the level of protein structure describing several connected polypeptide subunits. That is quaternary. Okay, primary is just your chain. Secondary is where you get your structures formed by stabilizing the chain into alpha helices, beta pleated sheets, and turns. Tertiary is your final fold, when you end up with your complete folded structure of whatever that polypeptide was, and you might be done at that point, but what's more likely is you connect up other subunits onto it, so each of those polypeptide chains, when they connect in, you've now got a quaternary structure. Okay. Region S has some of the R groups in contact with the bilayer. Predict the class of R groups to which these amino acids belong. Okay, so region S in here is all the way through the center of the bilayer, so they must be hydrophobic, because otherwise they would not be able to maintain contact with the hydrophobic lipid tails. Okay. It has been suggested that different forms of the GABA-A receptor subunit can arise as a result of alternative RNA splicing. Explain how alternative RNA splicing could result in the production of variant forms of GABA-A receptor subunits. Now, it's not far off a higher question here. Okay, so first off, alternative splicing means that you get alternative exons retained in code, retained, let's say, for translation. Okay, because you've got that, you will have different sequences of amino acids. So therefore different interactions. Different interactions means different shape, and always shape determines function, so their function also determines shape. It's a chicken and egg thing. Okay, suggest what happens to the receptor protein when GABA binds to it. Conformational change. This is your go-to. Anything which is making a protein change what it's doing has to be something which allows different parts of it to suddenly be able to do something, and the only way they can do that is if they can now come in contact, so it's always conformational change. The drug Diazepam increases the effect of GABA molecules by binding to a second allosteric binding site on the GABA-A receptors. State the term used to describe the effect of Diazepam on GABA-A receptors. Okay, so it says allosteric and it's increasing, so this is a positive modulator. Okay, and the positive modulators are also called activators. And because they've told you it's a drug and that it's increasing the natural response, they also have to accept if you said that was an agonist. And that's the question.